Welcome to the Hampshire and Isle of Wight Saliva Testing Laboratories. This facility has been designed by the University of Southampton and partners for the mass testing of the SARS-CoV-2 virus in human saliva. For more information about the programme itself, how to take your saliva sample, or for resources for parents and teachers and students, please visit the programme website. We're busy finishing off and adding final touches to the facility, but we thought we'd show you some of the features of our laboratories and explain the journey your saliva takes here. Throughout the building, it is important that everyone follows the one-way system, not only for the health and safety of individuals, but to also maintain safe operation within the facility itself. This building is divided into 6 different zones. These are physically separated and can only be accessed by following the green arrows on the floor in a one-way system. Zone A is responsible for the preparation of all material required throughout the saliva testing process for each of the zones. When plates and tubes are prepared, they are requested over walkie-talkies and delivered to each zone via trolleys. All samples are registered and tracked using a custom system based on barcodes. When a sample enters a new zone, its container's barcode is scanned so that we know where the sample has been and where it is going next. Saliva samples are picked up from each of the drop-off locations and delivered to this building, arriving at the rear on large pallets, which are emptied into a sample hopper. All samples are safely transported around the labs on either conveyor belts or trolleys. The samples arrive in 2 clear plastic bags, with the inner bag containing the sample tube and a leak detector strip. Sample bags are opened in Zone B on the ground level. The tube's barcode is scanned before placing it onto a conveyor belt, feeding them upstairs into Zone C, where the first stage of saliva extract happens. Once a sample tube has reached Zone C, it is placed into a custom-built robot which unscrews the tube's cap, extracts a small amount of saliva through a pipette, and deposits it into the well of an adjacent plate. This plate has a capacity to separately store extracts from 96 different samples. The plate is preloaded with a solution that, when heated, inactivates any trace of the virus if it's present in the deposited sample. Once the correct amount of saliva has been extracted and deposited into the plate, the robot screws the cap back onto the tube, ready for a technician to remove it and scan the barcode again. When a single plate has been filled with 96 different samples, it is removed by the white robot arm and sent to be heated. The tubes are stored in a box until we can confirm the results are negative. Afterwards, plates are collected and carried by trolley to Zone D. In Zone D, the plates of samples are placed into another robot, which extracts a small amount of a sample and deposits it in a tube of a genie strip. The genie strips are preloaded with a solution which reacts with the sample to help specifically identify the virus's genetic information by making it glow a certain colour. It is important that these genie strips are kept cold throughout the process, or else the solution deactivates, so we keep them in the freezer until the moment we have to use them. They are returned to the fridge until the next zone is ready to receive them for the final stage of processing. There are very strict rules for accessing Zone E. The strips are transferred to this room through a double-sided fridge so that people from the outside do not enter the zone and technicians working in the zone can obtain the samples without leaving. This minimises the contamination of samples which are now ready to be analysed. In Zone E, the genie strips are placed into a machine which analyses the samples by looking for a certain colour of fluorescent light given off by the reaction between the virus and the preloaded solution in the strips. If the machine detects a colour associated with the reaction with the virus, we can find out which saliva tube this came from by searching back through our barcode system. we can go back and find the original saliva tube and manually test the contents to confirm whether it definitely contains the virus. When we are sure that we can dispose of any boxes of tubes, they are emptied into a large oven along with all the plastic bags and steam cleaned at a high temperature so that they can be safely transported to a recycling facility. This is one way we are working to minimise the waste that we produce, as well as thorough sanitation of plates and other instruments to reuse in each zone. We follow strict health and safety guidelines at the Hampshire and Isle of Wight saliva testing labs, wearing all the necessary and correct PPE, as well as changing masks every 4 hours and adhering to the one-way system. If you have any questions or would like to see more details about the saliva testing program, how to take your saliva sample, or to see resources for teachers, students and parents, visit the Saliva Testing Program website.